welcome and thank you for taking out time to talk to us. Without further ado, I'll start, with our questions. So very basic. I would like to, talk about the basics. Tell us what is Sapient Bio and about the company basically.
Sure. And first, Himanshu, let me say thank you for the time and also to your organization, PharmaShots, for giving us this opportunity to share what we work on at Sapient. Sapient is a multi-omics company. We focus across production and interpretation of patient data in those different multi-omics, which are things like proteomics, lipidomics, and metabolomics. But we don't just produce lab data, we're really an insight engine. We give the biological context, the important patient context behind the data that we interpret. So it's quite frequent that we will put our own proprietary data assets or real world data assets into the analysis so that a pharma researcher or an academic researcher gets a much more full context of the disease state and how the patient would respond to therapy. We're really focused on moving the field of therapeutic research beyond the genome.
Thank you, Jonathan, for a very quick intro. So a little bit about if you can shed some light a little bit about the history of the company. When did you guys start? Maybe about the little bit about the co-founders, if you can.
Sure. So I joined only a few months ago, but the company's been producing and interpreting data since 2021, primarily in metabolomics and lipidomics. But since I joined, we have launched and are really focused on proteomics. It is a set of academics from the University of California, San Diego, that had some really groundbreaking technologies associated with how quickly and accurately you can analyze a patient sample. And we'll get into the details potentially in a little bit, but it is a more advanced form of mass spec, which is a technology that frankly has been around for quite a while. The difference is now in how we do it. Really, there are two. One, robotics and automation has raised the speed with which you can produce the lab data. And then especially the advances in informatics and machine learning have taken what was a very laborious and often error-prone process of taking data from the machines and interpreting it into an experimental result. We've reduced that time greatly. So overall, Sapient, our founders and our mission is all about the fast and accurate production of patient data, experimental data beyond the genome.
Awesome, great. So thank you for the history and obviously about the introduction. Now, because you mentioned about all the three omics, which is uh, metabolomics, lipidomics, and proteomics. So, and you're pushing beyond the genomic, the circle. So, can you explain why this is important for the advancement of the precision medicine? So, what's the application of all these omics and how it helps, precision medicine, which we are hearing a lot about nowadays?
Yeah, great question, Himanshu. I mean, just to baseline us, genomics is a great technology. It has been transformational for medicine. In genomics, you're looking at DNA, and then, I think through some very clever applications of genomics, the same machinery and the same approaches have been used for transcriptomics. And that means genomics is what DNA, what genes are present in a patient's cell. And transcriptomics is all about whether those genes are expressed and are on the way to become proteins. There, in biology, we have something called the central dogma, which is DNA becomes RNA, which becomes proteins. Genomics is a good technology for capturing the first two, the DNA and the RNA, and really understanding what is going on with those two. As we move beyond genomics, though, we're really very much focused on what happens in the cell beyond that RNA messenger. And this really gets to what is going on at the protein level and what is going on with a bunch of chemicals floating around in the cell or in the blood, things like lipids or metabolites that give a very accurate picture of the disease state for a patient. So when we say we're going beyond the genome, we're really focusing on what is actually happening in the cell, either in a healthy patient or in a disease state, where we can assess the proteins and we can assess other metabolites and lipids. And it gives a very accurate picture of disease that researchers use to develop new therapies.
Okay. Okay. While we are on the page of precision medicine, so can we dive a little more into precision therapy? So what is precision therapy? In your definition, basically, and what does how does Sapient play a role in in the drug development process of any precision medicine or a precision therapy, basically?
Yeah, great question. I mean, precision therapy is all about getting the right dose of the right medicine to the right patient at the right time. So that is the basis of precision. What that then means is that you really want to target certain disease cells, either specific tumors, or you want to get into certain organs, potentially like the liver or the pancreas. Or you want to ensure that the drug itself is only entering those areas of the body where it can have the greatest good and not have off-target effects or other organ effects. So as a result of all of that that is needed for precision medicine, you really need to look directly at your interaction with the drug target. And across all therapeutic modalities, small molecules, large molecules, biologics, et cetera, the protein is what you're drugging. And so it is the interaction with the protein that causes that therapeutic effect, that efficacious effect for the patient. It's also interactions with other proteins that can cause the safety issues, the toxicological events, the adverse events that cause a therapeutic to be either a limited label if it's approved or not to make it through clinical trials. So to do precision medicine, it is absolutely essential that you're able to accurately assess proteins, both the on-target proteins and their quantification, as well as other proteins that might have interactions with your developed therapeutic. It's really important also because I talked about the difference earlier between genomics and proteomics. And genomics, one branch of it, transcriptomics, which measures levels of gene expression, which should correlate with protein levels. Unfortunately, it often doesn't. And so to understand protein levels, which is ultimately what you are trying to drug, you really need proteomics. Looking at RNA levels, transcription levels is only a very vague way at looking what would otherwise be a very digital picture. So precision medicine, that precision is all about getting digital and quantitative with what is going on in the cell. And proteomics is the way to do that.
Okay. So, okay, so you say that proteomics is basically the key for precision therapy. That's I can maybe if I'm summarizing it in a right sense. Am I right?
Yes, that's right.
Okay, so now when we are talking about proteomics, how exactly Sapient helps Sapient helps drug developers to identify optimal targets with the help of proteomics.
Sure. Great question. I mean, let's take a step back and look at the pharma value chain. To develop a therapeutic, let me quickly take you through the steps that are needed. First, you have to identify a target, a drug target, some aspect of the patient's biology, usually a molecular pathway, that needs to be either fixed or disrupted because that's causing the disease state.
Okay.
When you have chosen a target, you then go in and try to develop a drug, discover a drug that will interact with that target. You identify a lead and then you optimize that lead. You often do that through high throughput screening. So then the result of those two, you now have a molecule of some kind, and you have something from the body that interacts with the drug target. You then go into preclinical testing, which is going to be testing in non-humans, often rat or non-human primates, or you'll do work with mouse models trying to better understand the disease. This enables you now to take that therapeutic, figure out what toxicological effects it might have, and to choose a dose. Once you've completed that, then you're ready for clinical trials. And a key success factor for clinical trials is being able to segment your patient population. So not all patients with a certain type of disease, but those that would really be able to benefit from your therapy. So you segment the patient population based on whether you think the drug will be particularly efficacious and particularly safe for those patients. And that leads to higher probability of success in clinical trials. So winding it back, Sapient and our work beyond the genome focused on proteomics and metabolomics helps with all of those phases. Through proteomics, we are able to identify new drug targets, and there are a number of companies that are very focused on novel targets because they have novel chemistry that interacts with those targets. We're also able to help in high throughput screening the selection of the therapies that would knock out those targets or interact with those targets. We're able to do that, as I was saying before, because we have radically raised the throughput and the time, the turnaround time from when we get a sample to when a result is available. So that means that our mass spec capabilities, our proteomics capabilities, are amenable to high throughput screening at that kind of pace.
Okay.
Further, our method of proteomics is not limited to humans. There are a lot of other technologies which you can only use with humans or you can only use with human plasma. We've developed our technology to be species agnostic, so it works just as well with a sample from a mouse, a rat, or a non-human primate as it does with humans. And that really helps with preclinical, really helps with toxicology, really helps with drug metabolism and pharmacokinetics, and especially really helps a company to select that dose for the clinic. And then lastly, as we get into clinical trials, we are a biomarker discovery company. So that enables us through either, often through metabolomics, or one of the few companies that does metabolomics, we are able to identify things that are going on in the cell that make a good signal to segment patients. And once you can segment patients reproducibly by something that is going on in their disease cells, then you are able to recruit the right patients into your clinical trial, those that will have a benefit from your therapy and not have a safety issue from potential toxicological events related to your therapy. That helps you to improve the probability of success for that therapy. So Sapient and our proteomics methods and our metabolomics methods help all the way across the value chain, from target ID, high throughput screening, preclinical development, and then ultimately clinical development.
So basically, when a client comes in or a pharma company comes in, Sapient is basically all the way through from preclinical to the to the clinical phase with them. So you are providing complete solution.
That's
Am I
right. We're
right?
one of the few companies that partner from early on, target identification or high throughput screening, all the
Yeah.
way through the clinic. And we have a CAP CLIA lab that we've developed specifically to be a partner for pharmaceutical companies in their clinical trials and even move beyond into diagnostics.
Awesome. So while we are talking about pharma companies and the role of proteomics, um, can you talk about maybe one or two actual case studies where uh, Sapient Bio have been have played a role, basically live case studies or actual cases where uh, you know and again, this is for proteomics. I am going to talk about the other omics, lipidomics and metabolomics also. But just for proteomics, if you can just share maybe one or two examples where Sapient played a very crucial low role, and client came up with a problem and then you provided a solution, something of those sorts.
Yeah, absolutely. If we first focus on proteomics, as I was mentioning, there's really been a revolution in chemistry, in therapeutic chemistry, in what's called medicinal chemistry. We've moved beyond a place where there used to just be a few hundred drug targets. And if you think about the fact that the human genome is 20,000 genes and all pharma research was focused on a small handful of that, like something under 1,000 drug targets. What has happened recently is the ability, some of it's AI, some of it's better chemistry, some of it is funding with the right sorts of companies. But overall, there's the rise of what's called a platform company. And a platform company is not formed necessarily to go after a specific disease or a specific biological pathway. They are formed because they're particularly good at the chemistry of interacting with a protein. And so, several of these companies exist saying we can drug anything. What protein should we drug? And so we work with those platform companies at scale to select the very best target for the disease state, which means look at the unmet medical need. You really want to focus on areas where patients do not already have a solution. And then you want to really understand that disease so that, much of the literature has been focused on the drug targets in that disease. So we partner with these companies not only to suggest a protein, but to look at how many patients then would benefit from a therapy based on that protein, how likely it is that drugging that protein will be safe and therefore have a higher probability of success in the clinic, and really bring our real world data assets to bear alongside our lab capabilities to give them a very complete understanding of that drug target before they launch into the most expensive parts, which are actually working on the chemistry, getting into the preclinical testing, and then ultimately moving into the clinic.
Okay. So, any again, my question is pretty much the same. Any pharma client you can name of where Sapient has or not just pharma client. I'm just saying any trial where Sapient supported in a proteomics assessment basically, and our audience can just go and check out that where, what stage, and all those kind of information. So if that is something which you can share, if not,
Yep,
that's okay.
absolutely. So we just talked about proteomics, so maybe I'll focus in on metabolomics. Now, just
Sure.
so, I think most of your audience is going to be very familiar with this, but just to set the stage a little bit. Metabolomics is able to, and our method for metabolomics, we're able to simultaneously measure 12,000 different analytes within a sample. So what does that mean? It means that there's lots of stuff floating around in your blood or in your cytoplasm that's never really been characterized. And biologists and discovery pharma researchers don't spend a lot of time or focus on these other molecules, because they're not drug targets and they're often not involved in disease. However, the levels of these metabolites can change radically based on the disease situation or the patient's response to therapy, critically the patient's response to therapy. So we've been able to discover metabolites as part of a clinical trial where we analyze the samples of participants, the patients in that trial, and we are able to tell really before the results come in which patients are going to benefit from therapy and which patients won't. And so what this means for a pharma company is that you can rescue a trial, You can do an adaptive clinical trial. You can change the protocol based on this new information. And you can take a drug that would otherwise fail and really rescue it in real time. So you discover the biomarker that's a metabolite, you observe the predictive power of that biomarker to predict patient response to therapy, either good or bad, right? It could be predictive efficacious, meaning the patient will benefit from therapy, or predictive safety, which means that maybe the patient's benefiting from the therapy, but there are a lot of other adverse events going on with the patient. Based on that, you can design or modify the protocol to enrich for patients that will benefit and to exclude patients that will not have a safe result. And therefore, you can refine your label, you can get a better approval faster, and overall rescue the drug. And we've had the opportunity to partner with a number of pharma companies in exactly that kind of biomarker discovery, clinical acceleration type project.
Great. So, if I understood it correctly, proteomics is basically as a technology. It could be used. It is more, used in the preclinical stage before the clinic starts, and whereas metabolomics is more into, which is more useful during the clinical stages. Correct me if I'm wrong.
Yeah, maybe just very briefly, those were two extremes that I gave you. I think that there's lots of interest in measuring proteins in the clinic. Proteins can be very good biomarkers. We've seen, interestingly, especially in rare disease, much more of an emphasis on approving therapies, not necessarily based directly on a patient state outcome, meaning does the patient respond to therapy in terms of condition improvement, but rather what's going on at the protein level. And you see this, as I say, fairly frequently now in rare disease, where you're directly measuring proteins as the outcome of the clinical trial. And I think you're going to be seeing more of this with precision therapeutics.
If I'm again, it's something which I was doing recently. So albuminuria is kind of, I think, a protein which you can assess, in a rare disease like C3G or IgA nephropathy, kind of, if I'm not wrong. Okay.
Yep, that's exactly right. All the way through from, as I was saying, rare disease, but all the way through to oncology. I mean, M protein levels in multiple myeloma, critical understanding for clinical success.
Awesome. Great. Now even I have understood. So thank you for that. Any example you want to, or a case study you want to talk about lipidomics, basically? I mean,
Yeah, yeah, just very, very briefly. I mean, you've heard sort of the broad sketch of other ways we use proteomics and metabolomics. Lipidomics is particularly important because in cardiometabolic therapy area, which is becoming a very large therapy area, incorporating everything from diabetes to obesity, all the way through traditional heart medications.
yeah.
There are some things that are really important in this type of trial or this type of therapeutic development. First of all, the number of patients in these trials tends to be much larger, especially as compared to a rare disease trial or an oncology trial. What that means is you have more patients, so you have a tighter patient response. You can, your statistics overall are better, but also you can discover some really interesting things going on at the lipid level. And so I think in the future, we are going to see a much deeper and nuanced understanding through lipidomics of the role of lipids in disease, but also the use of lipids to really segment patient populations, much like we were talking about in terms of metabolites or proteins. That field is still very much in its infancy, but some of the demographic changes in the US and across the world with aging populations, and the current state of obesity and weight gain really throughout the developed world, you're going to see this as a much, much bigger area of investment and pharmaceutical interest.
No, no, definitely, I agree with you because at the moment I cannot pinpoint the trial, but I have seen obesity trial which are assessing triglyceride levels in the as a biomarker. I would say in a very layman language, as a biomarker in a lot of trials, early stage trials. Plus, I have also observed that companies which are developing RNA medicines, RNA therapies, and even cell therapies or gene therapies, they are also assessing these kind of lipidomics and metabolites and proteomics, protein related biomarkers. So I completely agree with you. Uh, so I think yeah, thank you for that. And now just one very last question. And this is connected to the first reply which you have given to me. So probably we are doing the full circle basically. So you told me that you have recently joined. So as a new joiner and a person who is obviously handling the backbone of the company as a business officer, chief business officer. So uh, what's your vision for the company first? How do you see maybe I don't know how you want to define it, maybe short term goal or a long term goal kind of a structure, however you want to. And then the second part of this question is how does your past experience is going to benefit Sapient in the going forward journey?
Yep, great question, Himanshu. Let me start. First of all, what Sapient does. I mean, the two key things of where we're going. I've mentioned the high throughput capabilities that we have for highly accurate lab characterization of proteins, lipids, and metabolites. We've talked about that quite a bit during this call. What we haven't talked as much about, I think I mentioned it earlier, but we haven't really gotten into it, is our real world data asset. So we have 10,000 plus patients where we've sampled them across 10, 20, 30 years, plasma samples, and we've characterized those patients across proteomics, across metabolomics, across lipidomics, and now we're also sequencing them across genomics. And so it's a very deep data set with patient outcomes. So, what was the response to therapy? What therapies were they on? Um, and what underlying demographics, ethnicity, and treatment courses did they follow? So, a very deep data set. So, what I'm really focused on now and where Sapient is going, we will continue to do lab characterization, but our goal is to bring much more biological insight in not just the raw mass spec data file that we dump back on our client, really partner with the client to give a biological insight that's informed by our real world data, our deep characterization of those patient samples, and then what we found from the samples in the experiment that we did for the sponsor. And so we put those three things together. I think we're really going to have much more acceleration in therapeutic development and really go after serious unmet medical need for patients.
Awesome. I mean, RWE and real world data is something which I am also very interested in. I've been, reading about it more frequently than I ever thought and imagined. And obviously, today only we have 30 minutes. Your schedule is busy. I understand that. So maybe we should catch up sometime again and talk a little more about real world evidences and what all new things Sapient is doing. So thank you once again, Jonathan, for your time. Thank you, Carla, for facilitating this for us. And I hope to catch up with you again and have the same interesting conversation with you again, and probably the bigger team of Sapiens.
Thank you very much, Himanshu. I mean, I really enjoyed the conversation. I think, we really got into some of the details, and I would really welcome the chance to talk to you again.